There's a question that's posed by those who seek new knowledge. What is possible, they ask? What avenues might we walk that have not yet been traveled? We call them scientists, but they're discoverers, really. The eloquence of their discoveries is expressed in the equations and formulas that record their progress. In a way, these symbols represent a language all their own, the language of possibilities, the language of research. It's a language that gives voice to a deeply held belief common to so many of us, a belief that within the infinite possibilities we call science can be found the means to improve the human condition. It's the pursuit of this belief that calls upon the creativity, curiosity, and inventiveness of individuals who transform what might be into what is. This year marks the 100th anniversary of an exceptional commitment to exploring these realms of possibility, the Lilly Research Laboratories. It was in 1886 that Lilly Research Laboratories began. That year, Colonel Lilly hired Ernest G. Eberhard to operate the newly created research laboratory. The eagerness to create a research effort was in part a product of the times. The late 1800s were years rich with invention. Bell, Edison, and Marconi were all at work in their own labs, and the world was ready to awaken to the exciting possibilities of new technologies. Admittedly, the earliest tasks of the fledgling laboratory were very modest in their intent. There was a strong emphasis on product uniformity and upon monitoring the process of manufacture. But even this devotion to practicality is very important to note, because if there was one vision that Colonel Lilly held for his company, it was to manufacture products of the highest quality, products that were also consistently reliable. This notion in and of itself was quite remarkable in an era dominated by patent medicines and snake oil salesmen. Quality was the Colonel's hallmark, and it has remained a central tenet to this very day, continually influencing the direction of the company. However, controlling quality in those early days could become something of an adventure. There was, for example, a rather troublesome incident concerning a batch of extract of thimbleweed that had been returned by a customer. J.K. Sr. and Eberhard decided to test the extract themselves, and within hours, J.K. Sr. was found lying unconscious by his desk. Fortunately, a doctor was able to revive him. As it turned out, a poisonous variety of thimbleweed had been used accidentally in preparing the extract. Products like extract of thimbleweed reflected the times. It was largely an era of botanical products, of tinctures and extracts, elixirs and potions. And indeed, the research laboratories at Lilly were actively engaged in creating and developing botanicals. Lilly products such as cocoa quinine and ipecac became household names. Meanwhile, science was creating a new age, one that would launch medical research on a path rich in new discoveries. The works of Louis Pasteur in France and Robert Koch in Germany in the late 1800s demonstrated the causal relationship between bacteria and disease, and resulted in the first successful vaccines. The new possibilities that this work created represented a new era of biologically based products. And as the Lilly scientists turned their attention toward the study of living things, they began to discover an expanding array of vaccines and antitoxins. In 1913, the company established its own biological division. A year later, the greenfield operations began. 156 acres set up to house the hundreds of animals that served as living factories to produce the new biological products. Smallpox vaccine, diphtheria vaccine, influenza vaccine, rabies vaccine, each meeting age-old medical needs with the new biological technology. As biological investigation intensified, certain substances manufactured within the body began to attract attention. They were hormones, the powerful agents of change within living things. In 1919, J.K. Sr. established the Lilly Glandular Research Laboratory. and Lilly scientists began to investigate the role of hormones. The early Lilly scientists focused much of their attention on developing glandular products, such as various pituitary extracts, thyroid extracts, and desiccated parathyroid products. As the 1920s approached, glandular and biological research activities in medical science were increasing rapidly, and the company realized that in order to stay abreast of this wealth of knowledge, it needed a special type of individual. To meet this need, the company chose a British-born chemist by the name of George Henry Alexander Clues. The year was 1919, and within a short time of his arrival at Lilly, he helped establish several research programs with educational institutions. But by far the most historic relationship that Clues forged was with Frederick Banting and Charles Best of the University of Toronto, because it was from this relationship that the company embarked upon a mission of major significance: to create insulin on a scale large enough to supply the needs of diabetics throughout the world. If the mission were successful, the impact on human life would be nothing less than phenomenal. The company's previous work in glandular research had paved the way for this undertaking. However, the difficulties of refining, processing, and then producing this newly discovered life-giving substance were still overwhelming. Through a concerted effort to explore every available avenue of possibility, the necessary technology was refined, enhanced, and made practical. There were many individuals who made highly significant contributions to the development of insulin. One was George Walden, who was instrumental in developing a workable method of obtaining the active substance of the pancreas through isoelectric precipitation. And in October of 1923, a breakthrough product, Lilly insulin, known as Ileton, reached the marketplace. It's the curious nature of research that each new answer spawns its own set of questions. And so it was that, at Lilly, insulin research didn't stop with Ileton. There were more possibilities to explore, more needs to fill, and ultimately, a wide range of insulin products would be created. In 1926, the glandular research program was engaged in work involving liver extracts. Lilly scientist Edward Campbell was working with liver extracts as treatments for hypertension. At the same time, in Boston, George Minot and William Murphy had developed a treatment for pernicious anemia, feeding raw liver to their patients. And although the treatment was effective, it had a major drawback. Most patients couldn't maintain the treatment without becoming seriously nauseated. In Indianapolis, Campbell, along with Dr. Leon Zerfus, began some successful experiments using their liver extracts as a treatment for pernicious anemia. One of the patients who received these experimental treatments was Mrs. J.K. Lilly Sr. At that time, pernicious anemia was almost always fatal within 2 years of diagnosis. But with the help of the experimental Lilly treatment, Mrs. J.K. Lilly Sr. lived a full 8 years after her disease was diagnosed. Shortly after the Lilly experimental work began, Dr. Minot visited Indianapolis, and a path was opened for a collaborative effort between the Harvard and Lilly scientists. The result in 1928 was a liver extract product, a dramatic new treatment that greatly extended the lives of those suffering from pernicious anemia. Through its development of Ileton, the company had learned the value that could be gained from the controlled testing of its products. At the time of its introduction, Ileton had been more extensively tested than any other pharmaceutical product of the time. Also, the development of an effective treatment for pernicious anemia virtually necessitated the help of human subjects. So, in order to satisfy this growing need to understand precisely what the effects of new medicines would be, a rather remarkable arrangement was reached. In 1926, a Lilly research clinic was established in Indianapolis at what was then called City Hospital. The Lilly Research Clinic offered a clinical laboratory for product testing and development, as well as a controlled environment in which to conduct research in humans. The clinic's longtime head, Dr. Kenneth Kolstad, was instrumental in building the clinic's stature with academic centers throughout the world, a tradition which has been carried on by each of his successors. In much the same way that there was a need for clinical trials, there was also a growing need for a more comprehensive understanding of the precise effects that drug substances have on living cells. It was the beginning of an era of pharmacology. Once again, the company went out in search of its future, in search of individuals who could help to usher in this new age of research. One of those who personified the growing pharmacological orientation within Lilly Research was Dr. K.K. Chen. Originally of Shanghai, Dr. Chen had already been in contact with Eli Lilly and Company. While still in China, his work had led to the Lilly product ephedrine, an effective asthma treatment. Later, while Dr. Chen was conducting research at Johns Hopkins, J.K. Lilly Sr. invited him to join the company. As early as 1925, the company had developed and released its first barbiturate product, Amitol, but there was still a great medical need to find an effective means of alleviating pain without relying on morphine-based substances. Under Dr. Chen's direction, research in sedatives and analgesics was expanded, and in 1936, Seconal, a new barbiturate of shorter duration, was developed. A corollary to the growth of pharmacological research was the investigation of the adverse effects that drugs might have on living cells, the science of toxicology. Indeed, Robert Anderson and other Lilly scientists had conducted a number of toxicological studies in the early 30s as an outgrowth of their ongoing pharmacological research. But it was a series of tragedies in 1937 involving a drug by another manufacturer that really galvanized the company's efforts in toxicological research. The Lilly Research Laboratories increased its efforts in the study of long-term toxicity and established an ongoing program devoted to toxicology. Today, the analysis of a substance's potential toxic effects extends over generations of animals. As the Lilly scientists entered the 1940s, they became involved in an area of research that would forever change medical science. In a way, it can be traced back to London, England in 1928, when Alexander Fleming first noticed some curious germ-killing properties of a green mold on one of his culture plates. The mold was Penicillium notatum, but Fleming's observation remained largely overlooked. Overlooked, that is, until the rising thunder of World War II created an anguished cry for an effective means of fighting infection. Howard Florey, working at Oxford, found a way to refine small amounts of penicillin, but it was left to the resources and ingenuity of the American pharmaceutical industry to find an effective way to mass-produce the new wonder drug. Under government auspices and wartime priorities, several pharmaceutical companies set about the task, each trying a slightly different approach. At Lilly, the penicillin research effort was nothing short of dramatic. James McGuire had started some penicillin experiments when Florey's initial studies had been published. The United States Surgeon General officially asked Eli Lilly and Company to work on the problem. The effort expanded rapidly, and in less than a year, the research group went from growing penicillin in glass milk bottles to fermenting it in 3,000-gallon tanks. John Leighty, George Walden, and Harley Rodehamel Jr., along with many others, played significant roles in the production of penicillin. But the story didn't stop with the first tankful of what was then the most potent antibiotic known to humanity. Lilly scientists continued to look at penicillin and to search for new possibilities, better ways to refine, purify, and modify. And their efforts led to important new discoveries. A longer-lasting form of penicillin G, known as procaine, was developed by Rodehamel and Leighty and introduced as duracillin. This was followed by the development of orally active penicillin V by Otto Behrens and Reuben Jones. Penicillin paved the way for a new age of pharmaceuticals as scientists around the world searched to discover more miracle drugs. At Lilly, a program of investigating soil samples from around the world was begun. One sample from the island of Iloilo in the Philippines yielded a new antibiotic, erythromycin. In 1958, the Lilly Research Laboratories embarked upon a course of antibiotic research that would eventually lead to discoveries of the highest consequence. Building upon the initial work done by Professor Abraham and Dr. Newton at Oxford, Lilly scientists pioneered the creation of an ever-expanding family of cephalosporin antibiotics. Today, cephalosporins are the most widely used class of antibiotics in the world. Shortly after the early antibiotic research began, an intriguing new medical technology captured the interest of Lilly scientists, that of tissue cultures. At the time, in the late 40s, there was no immediate application of this technology to the development of a new product, but it did yield valuable insights into the ways that viruses reproduce. The promise of this research was not to be realized until the early 1950s, when a disease called poliomyelitis raged throughout the world, crippling and killing people by the thousands, particularly young children. When in 1953 Dr. Jonas Salk announced his work on the development of an effective vaccine, the manufacturing companies moved rapidly toward production. There were, however, decisions to be made. The Lilly scientists, led by Doctors Clyde Culbertson and Herman Detweiler, chose a method they felt best for culturing the difficult-to-kill polio virus. Other manufacturers made other choices, and there were problems. Ultimately, the Lilly vaccine was found to be pure and, more importantly, safe. Eli Lilly and Company is credited with supplying well over 50% of the Salk vaccine, and today, few cases of polio exist in the world. As the company moved through the 50s, scientists began to consider the ever-widening scope of knowledge we call the life sciences. All living things, whether plant, animal, or human, are biological systems that respond to various compounds in specific ways. Why restrict the Lilly laboratories to humans? And so it was that the Lilly scientists began to apply their thoughts to agriculture. In 1954, a new area of business was born, the Agricultural and Industrial Products Division. And scientists considered the possibilities. Might it not be possible, they wondered, to protect crops and animals and improve upon their rate of growth and productivity? As has been the case so many times before, once the possibilities were explored, discoveries were made. It has been said that the single most significant event of our times was the discovery of the molecular structure of DNA. And certainly, that discovery has created a profound impact upon the direction of research at the Lilly laboratories. It may be, for example, that there exists a link between our genetic makeup and our susceptibility to disease, and scientists are beginning to cast their scrutiny inward toward the body's own systems. Within Lilly Research Laboratories, an intense interest in the human immunological system has developed. Perhaps the scientists reasoned, through a greater understanding of the body's ability to counteract disease, important new medical technologies can be developed. Scientists speculated that the natural proteins within our bodies could be duplicated and recombinant DNA products could be developed. From this premise, a wealth of new possibilities are being revealed. For example, Humulin, the first commercially available human healthcare product produced by recombinant DNA technology. Proinsulin, which may help to reduce serious complications and significantly improve the lives of diabetics. And Humatrope, a biosynthetic human growth hormone, chemically and structurally identical to growth hormones made by the body. Lilly scientists are investigating a genetically modified tissue plasminogen activator, TPA, which may be valuable in the treatment of strokes, phlebitis, and heart attacks. and APC, activated human protein C, a plasma protein that could serve as an anticoagulant during major surgeries. Today, Lilly research activities literally span the globe, from the fine old Spanish-style buildings of Greenfield to the Lilly Research Center at Earlwood near Windlesham, England, to the clinical tradition at Wishart Hospital in Indianapolis, to research laboratories in La Jolla and throughout the world. Lilly scientists are also encouraged to attend and participate in symposia to hear and share ideas in the most timely way possible. And Lilly continues to encourage the examination of new possibilities through the sponsorship of research programs at major universities and research laboratories throughout the world. The ongoing tradition of research at Lilly has extended to each of the 4 medical instrument companies and to the research programs at Elizabeth Arden and now at Hybritech. The commitment to research is indeed substantial. Approximately 4,000 people, or 1 out of every 7 Lilly employees, is engaged in research. And this year, over $400 million will be spent on research activities. As always, as new discoveries are made, new possibilities are revealed. But there are some things that remain constant. A document prepared in 1916 by J.K. Lilly Jr. states in a simple but direct way a basic value that is as much a part of the company today as it was then. It's a sincere concern for each individual in the company and a philosophy of encouraging individuals to reach toward their own potential and to explore their own possibilities. As Mr. Eli said, build men, then medicines. When this attitude is combined with the Colonel's original vision of creating products of the highest quality, a fertile environment for research is created, one that fosters the creativity and ingenuity of the many scientists who have built this tradition. Yet in the long run, perhaps the greatest contribution of values isn't the atmosphere they create as much as it is the continuity they provide, a continuity for the future. In the most important of ways, the future of Lilly Research Laboratories carries an important value from the past, a continued commitment to travel down the avenues of discovery, searching for new ways of giving expression to the language of possibilities. For new ways of giving expression to the language of possibilities.